Today we'll talk about the fundamental tissue and mitosis. We will have three parts of this. The first part has to do with a virtual microscope, how to identify things from the images by using the computer. Describe the steps in tissue processing and discuss the different types of microscopy, both light and electron microscopy. Then we will interpret cytologic features of cells by different staining, differential staining, with hematoxylin and eosin are main stain, but we also use PAS and toluidine blue, as well as processing tissue for conventional transmission electron microscopy as well as scanning electron microscopy. Part 2 has to do with recognizing and identifying cells using the microscopic preparations that we have, the images that we have, a little bit about how to observe the different structures in the images that we have, and to understand the relationship between structure and function of cells. Part 3 has to do with mitosis, talk about the stages of mitosis and cellular structures that are associated with mitosis. Mitosis is a process of cell division by which you produce two daughter cells identical to the previous parent cell. Enjoy. Fundamental tissues and mitosis. This is part 2. We'll be talking about the images of light and electron microscopy. So we want to recognize, identify cells in microscopic tissue preparations and to understand that morphology reflects the functions of cells is what we want to talk about. So in this lab experience, we want to gain a greater appreciation for similarities and differences of cells and tissues, and we want to be able to interpret them as how it relates to overall structure, and we want to remember to use your atlas. Atlas is a very handy thing to be able to use to identify things in the textbook, to be able to review some things. Here on slide 29 we have skin, and the upper part in through here is dark staining is epithelium. It's composed of epithelial cells. This is the epidermis. In contrast, below that, the lighter staining material, which is mostly collagen that we see, collagen fibers, is the dermis, epidermis and dermis of skin, as we can see. Again, we can see it. This is the epidermis, and you can see there's different layers. There's those on the base, there's the spinosum, granulosum, and then the stratum corneum at the top. These are different layers. Don't worry about identifying those now. Just to say all these are epithelial cells. They are produced in through here by mitosis, and then they differentiate as they go to the surface and finally are removed, sloughed off. The FBI are looking for those if they're looking for you. And then what we see here is collagen fibers. So there's a fibroblast nuclei, but in between there are the extracellular collagen fibers that we see. Here's a tongue. We can see some different things. There's skeletal muscle that we see through there. There's also serous glands. So this is a glandular structure. We can see these dark things here are serous glands. And there's adipocytes. These are fat cells that are located in through there. So we have skeletal muscle. Since this is tongue, we need skeletal muscle where we can control it, and these are long cells with the nuclei on the periphery. Glands, so these are some of the glandular secretions of the saliva that we have, as well as some fat cells that we have in through there. Again, we can see a higher magnification. This is one of the acins of these cells. They secrete their contents into the lumen in through there. So it's just serous glands. Serous mean they're proteinaceous as opposed to a mucus type secretion. Serous glands have a spherical nuclei located pretty much at the base, but they're not flattened like mucus cells. Adipocytes, this is actually pulled apart a little bit, but these are individual cells in through there. Adipocytes, and then there's a skeletal muscle, as we talked about before, to be able to control movement of the tongue. Here's the kidney. The kidney shows convoluted tubules. This is the cortex of the kidney and the medulla of the kidney. If you look at where that little box is there, The box is illustrating what we're seeing on the overall specimen. So if you look right there, we're looking in the cortex. In the cortex, we have the renal corpuscle. We have lots of proximal tubules. Most of those here are proximal tubules. However, we do have a distal tubule that goes through there occasionally, that we see a distal tubule as well. But in preparation of the specimen to see it, you can see the magnification. This is the identification. You also can see a magnification if it's happened to be there. But you can move this around with your pointer, and that will make this image move. Again, we're in this little box right in through there, and we can see that this is a thyroid gland, and this is a thyroid follicle. In the follicle, you have simple cuboidal, that is one layer of cube cells that surround it and produce a colloid. We have extracellular collagen. All collagen extracellular is produced and is on the outside of cells. Collagen is mostly non-pigmented. It's the white of the eye that you see is the collagen fibers. The blood vessels, this is like probably a vein with a big lumen. There's some white blood cells and red blood cells in through there. This is maybe a venule. And sometimes we can see little capillaries in through here and here. A little artery right there. It's kind of wrapped apart, but that's a little artery there. And you have simple squamous, simple one layer of cells lining blood vessels known as endothelium. Here's a pancreas, and these cells more pyramid shaped, so they'd be more like maybe cuboidal, but they're certainly pyramid shape. Here's a group of them. On the base you see the nucleus, but also see the blue cytoplasm of the cell. In the apex near the lumen, we can't see the lumen. This is a cell in the lumen, nucleus of a cell starting a duct in the lumen, so this would be the lumen right in through there, and the red granules are actually secretion. So hematoxylin is staining the nucleus dark, and the base cytoplasm, which is rough endoplasmic reticulum, dark, ribosomes thereon, and then the red hematoxylin is staining the zymogen granules. So you see the base blue, nucleus blue, apex is protinaceous, and hence and here we can see the individual cell rough endoplasmic reticulum of this blue and through there. That's what we're seeing. There's a nucleus, and then the zymogen granules are the secretions of the cell that we see on its surface. Endoplasmic reticulum is stacks of sheets with ribosomes on its surface. You can see how ribosomes spot the surface of this. This is actually a nuclear envelope, and the endoplasmic reticulum, rough endoplasmic reticulum or granular endoplasmic reticulum, is continuous with the nuclear envelope. In fact, you even see ribosomes on the outer leaflet of the nuclear envelope. Nucleus, rough endoplasmic reticulum, more rough endoplasmic reticulum or granular type because it has ribosomes on its surface. Nucleus, nucleolus, nuclear pores, mitochondria that we see there. Here's more granular or rough endoplasmic reticulum that we see there. There's also some free ribosomes there. There's glycogen storage here, as you see there too. So this is granular and agranular. Agranular is smooth endoplasmic reticulum, characteristic of steroid cells. And you can see that these cells have lamellar cristae. The mitochondria have lamellar cristae, in contrast to steroid secreting cells. The cristae are more tubular type, tubes, as opposed to lamellar structures that we see. If you look at the plasma membrane, and we see two plasma membranes here, there's one from one cell and one from another cell, and you see they have a trilambar structure. They have two black lines and a white line in the middle. And this shows you where the light area in the center that we see, not here but in between here, is the phospholipid. So this is the lipid that doesn't like water. It's hydrophobic in contrast to the hydrophilic region. The phosphate heads takes up the stain. So osmium is positive along the phosphate heads, but not in the fat part, and that's why you have the trilambar structure that we see there. Also, if you actually pull back the top part, this is the E face or the ectoplasm outside, or the P face, which has more particles in it, which is the protoplasmic phase, the part that touches the cytoplasm. E touches the air, P touches the cytoplasm or the protoplasmic phase. And we can see those again. This is the P phase, lots of particles, and the E phase of a membrane. And we can remember, so this part here is pulled back, E phase, and the P phase with all the particles next to the cytoplasm. Here's another. So this is a carbon replica. So you don't really have the real specimen there. You have the replica that we talked about earlier. And here's another carbon replica showing you nuclear pores. So this is the nucleus in the cytoplasm. You see the Golgi, vacuole, and some more of the organelles that are located in through there. But the carbon replica lets you see the amounts of nuclear pores that we see. So in terms of the plasma membrane, you see the plasma membrane is what lines or limits the cell. This happens to have, this is a little capillary, and so you can see it makes a lumen. So the plasma membrane actually makes a lumen with junction between adjacent cells there. This is muscle, and around muscle these are vesicles that are inside the muscle cell itself. And then this is endothelial cells. You can see where you have transport of things through the vesicles from in the capillary lumen to the outside, as you see. And this is a penecytic vesicle, which usually is associated with tasting the environment. So here we see mitochondria here, here, and here. This is smooth endoplasmic reticulum here, rough endoplasmic reticulum there, and some smooth endoplasmic reticulum located in through there. These are lysosomes. There's actually secondary lysosomes that we have here. Mitochondria could have tubular cristae, and they have more than one membrane, so double membrane. There's mitochondria here and mitochondria that we see there. This is lipofuscin or mitochondria. You have different chambers. You have a chamber on the inside which has the electron transport system to produce ATP, the purpose of mitochondria. And anywhere you need it in the cell, like a ciliated cell at the very top where you have cilia beating to generate energy, you have a lot of mitochondria. Muscle cells have a lot of mitochondria, especially big ones in cardiac muscle cells. Also for transport, membrane transport, you have a lot of mitochondria, like in parietal cells, which produce hydrochloric acid to pump the hydrogen and the chloride ions out. They need a lot of mitochondria that's through there. Sometimes mitochondria look very similar to microbodies. Microbodies, as a rule, don't have as many of these things, which appear to be the cristae that we have in mitochondria. And also the mitochondria are double membrane, which microbodies should be a single membrane, as we can see. Golgi could be profile like this. It could be a profile like this. Pancakes in through there, or smooth endoplasmic reticulum, rough endoplasmic reticulum, mitochondria, and the nucleus. And there's another nucleus here as well, and this is heterochromatin, euchromatin in the nucleus. Lipid droplets, you can see lipid droplets. It's in intestinal absorptive cells, which has a brush border. Uniform microvili is what makes a brush border, by all the same length. And here again we have secretory granules coming out from the Golgi apparatus. Again, the Golgi apparatus. Glycogen looks very similar to ribosomes, but a little bit larger, and usually forms in a rosette. And here we have lipofuscin, or a secondary lysosome. Now the mitochondria, and this is microtubules. Microtubules, very long microtubules. Here we see microtubules here associated with chromosomes pulling apart, and also there's microtubules here as well as part of a centriole. These are stable microtubules. These and these are labile microtubules, a breakdown. Here we see the axoneme, which is a 9+2 microtubule, 9 doublets plus a central pair. And you see the plasma membrane around. These is cut through some sperm tails at the end, where we be able to see the axoneme in each one of them. Cytoskeletal components, microvili, actually project up and take the plasma membrane with them. So microfilaments projecting up inside, pushing the membrane out, is what makes the microvili. And if you see a cross section of these, you can see that these are indeed the microfilaments. Down in through there, you got the terminal web, which is what you see here. This is a plasma membrane, and these are the same as those, and you can see that the microfilaments project up through and through there and push in the cytoplasm along and make these. Also note on the outside of this, we see the trilaminar structure again. On the outside, we see a fuzzy coat. You see the fuzzy coat right here. Remember that fuzzy coat is really carbohydrate rich. Remember the PAS staining of the fuzzy coat is actually the carbohydrates on the surface that are staining. So in terms of clinical correlation, we have one, apoptosis, which is a programmed cell death, like maybe a tadpole changed to a frog. You have cells that die part of the process. Necrosis, however, is different. Things just look like they just deteriorating. Rotting is a way that they look like. Apoptosis, you have organized thing where the DNA, if you looked at the DNA, it would have a ladder. It would have little bits of DNA that's broken in a programmed way. Not so for necrosis. So that's it. Thank you.